So fructose 2,6-bisphosphate is a regulator of phosphofructokinase 1. I talked about that last time. And I wanted to mention a little bit about how it's synthesized. Your book actually does a pretty good job, although it's a little bit confusing, how we can, how this small molecule, relatively small molecule, can get higher or lower inside the cell. And just to remind you, remember that this molecule is right here, this fructose 2,6-bisphosphate. It causes an increase in this activity of phosphofructokinase. Okay, so it turns out, just by looking at your book, that there's this enzyme, excuse me, that makes this this product, this fructose 2,6-bisphosphate, which turns on glycolysis. And the name of the enzyme is PFK2. It's right here, It's PFK2. So don't forget, PFK2 is the one that makes this fructose 2,6-bisphosphate. And it turns out that glucagon, okay, is when you get high levels of glucagon, it's kind of actually there's a little bit of a confusing diagram. What happens is that this kinase actually gets deactivated. It gets deactivated, and it gets phosphorylated. So the kinase gets phosphorylated. This little P represents a phosphoryl group. And now what happens is that you actually start to destroy the fructose 2,6-bisphosphate, and so the fructose 2,6-bisphosphate decreases. Now let me think about this. The fructose 2,6-bisphosphate decreases, and so what's that going to do to glycolysis? What's it going to do to glycolysis when the fructose 2,6-bisphosphate decreases? Let's go ahead and cheat. Look at this. Okay, now you have low levels of fructose 2,6-bisphosphate. What's it going to do to this pathway going to breaking down glucose? Remember, we're decreasing the amount of fructose 2,6-bisphosphate. So it's going to inhibit glycolysis, right? So it's going to inhibit glycolysis. Okay, so that's exactly right. It's going to inhibit glycolysis, and this is exactly what you want to happen in the liver. So recall, I'll draw my diagram of the liver. There it is, my professional diagram of the liver. That's the liver, okay? And so in times of starvation, glucagon gets secreted from the pancreas. I'll draw the little pancreas here. There's the pancreas, okay? And the glucagon late at night, when you're tossing and turning at night because you haven't had enough to eat, the glucagon's going to go into the bloodstream and bind to the surface of the liver. And what this does is that this eventually leads to a decrease in glycolysis. Okay? And there's a reason for this. And the reason for this is that the liver, as I said before, is the master supplier of glucose to the rest of the body. The rest of the body, the muscles, okay, so here's some muscle cells, okay, here's the brain, okay, that's the brain here. These are starving for glucose, okay, so they're very unhappy, okay, so these guys are unhappy, and so what happens is that glucagon gets secreted from the pancreas, it shuts down glycolysis, it's going to increase gluconeogenesis. Okay. It also increases glycogenolysis, which I didn't talk about too much, but that's the breakdown of glycogen. Okay? And that way you have a good supply of glucose here, and that glucose then can be exported into the blood so that now these muscles and the brain can be happy and they can survive. Okay, so this liver is really important for that. As a matter of fact, there's a diagram that draws these things slightly better for you. Okay. And it's on page 759 of your book. I know that's ahead, but I want to drive home this point of the exquisite regulation of glycolysis and gluconeogenesis. So here we have the pancreas here. It's a better pancreas than mine, I admit it. Okay, and then here's the low blood glucose, okay, and that signals glucagon to be secreted, okay, and then now we have high levels of glucose in the liver, and now the liver will then export glucose into the blood. Ignore this blue line for now. That's about insulin, but just concentrate on the pink line here. So this is on page 759 of your book. Okay. So this enzyme then shuts down. It doesn't make any more 2,6-bisphospho fructose. 2,6-bisphosphoglucose. 2,6-bisphosphofructose. I'm sorry. Okay. So that's great. That's wonderful. Okay. And then just to complete the story, when insulin levels are high, so after you've just had a meal, okay, what happens is that now you convert this enzyme that makes fructose 2,6-bisphosphate into its active form, and now you start to make more of this fructose 2,6-bisphosphate in the liver, okay? And that's good, because now the liver can use that glucose for its own metabolism. It can start catabolizing the glucose. This is a very interesting enzyme. It's unusual because it actually has dual function. It's a kinase as well as it's a phosphatase. So it has actually two enzymatic functions on one polypeptide, and it's regulated by phosphorylation. Okay. Well, I want to talk a little bit about another organ, okay? And this other organ is the heart. Okay, so let's talk about the heart. Okay. Okay, there's my heart, okay? And in the heart, what happens is that when you get, before I do that, before I go to the heart, hold on, let me just talk a little bit more about how this glucagon works at the molecular level. So glucagon is a polypeptide secreted from the pancreas, let me see, the alpha cells in the pancreas, okay? And what it does is it binds to the liver. I'm going to just draw a liver cell. So this is a liver cell. Okay. And there's a special receptor called the glucagon receptor that is a membrane protein. You guys have studied membrane proteins. That sits on the plasma membrane of the liver cell. So glucagon will bind to that, okay? And upon binding to this, there is something called a G protein, and that G protein gets activated, and it slides along the plasma membrane on the inner surface of the plasma membrane, and it turns on this enzyme known as adenylyl cyclase. Okay? And what adenylyl cyclase does is it converts ATP to something called cyclic AMP. and what leaves is something called pyrophosphate, which I'll abbreviate as PPI. That cyclic AMP will bind to a kinase known as protein kinase A. Okay, binds to protein kinase A. And protein kinase A then will go ahead and phosphorylate. It's going to phosphorylate this, this enzyme known as PFK, PFK2. And so that's how this whole glucagon thing works. That's how the glucagon thing works. We call the cyclic AMP, we call that a second messenger. Okay. And this whole scenario here where glucagon binds this protein, activates this G protein, which activates this adenyl cyclase, that whole concept here, this whole thing is called a signaling pathway. That's called a signaling pathway. And our body is filled. Our cells are filled with these signaling pathways that cause certain responses to hormones and small molecules. So that's how this protein phosphofructokinase 2 gets regulated. All right. What about the heart? Okay. In the heart, you also have this phosphofructokinase 2. Keep on saying PK. Phosphofructokinase 2. Okay. But when you have low energy charge, okay, what happens is that this phosphofructokinase 2 gets activated. And it gets activated differently than it does in the liver, okay? And what happens is that when you have low EC, you have high levels of AMP, okay? Get high levels of AMP. And that those high levels of AMP will bind to an enzyme known as AMP-dependent kinase. Okay? And that enzyme, the AMP-dependent kinase, will then go ahead and phosphorylate this phosphofructokinase 2. And once it phosphorylates it, it actually activates the phosphofructokinase 2. So now the phosphofructokinase 2 gets activated. I'm going to say activated. Okay. Activated phosphofructokinase 2. Okay. And now we're going to be able to make high levels of fructose 2,6 bisphosphate. Okay. And then what does that do to glycolysis? Is it going to activate glycolysis? Someone said yes. It's exactly right. It's going to activate glycolysis. So that's exactly right. So you can see that this phosphofructokinase 2 is very important depending on what phosphorylates it. If it's phosphorylated by protein kinase A, PKA, then it becomes deactivated. Okay? If it's phosphorylated by AMP-dependent kinase, then it becomes activated. Let me show you again a couple of images from the from your book. Okay. This is an amazing enzyme, this AMP-dependent kinase, because it responds to low energy charge. Okay. So this is on page 762 of your book. And it's a very complicated diagram, and I only want you to concentrate on just a few things in here, because it's very easy to get dizzy looking at this type of diagram. I just want you to look at this. I've written here in pencil low EC, low energy charge. What that means is that the AMP to ATP ratio has increased, okay? It binds to, AMP will bind to this kinase. This is an enzyme, okay? And once that enzyme binds to kinase, it's going to phosphorylate. It's going to phosphorylate this enzyme known as phosphofructokinase 2, okay? And when that happens, you get high levels of fructose 2,6-bisphosphate, and then you get an increase in glycolysis. So this is what's happening in the heart, okay? And other organs as well, but also in the heart, okay? All right. In the liver, I'll just go to another page in this chapter 18. In the liver, this is exactly what I just said. We have something else happening. We have glucagon, as I said, being secreted from the pancreas. It's going to create high levels of cyclic AMP in the liver cell. One of the enzymes that gets activated is protein kinase A. So I've written down here pKa. Don't get that mixed up with the classical pKa you guys learned in general chemistry. Okay, this is protein kinase A. Protein kinase A will then phosphorylate this phosphofructose kinase 2, okay? And in doing that, it converts it to actually a phosphatase, okay? And what happens is that you get lower levels of fructose 2,6-bisphosphate. And when that happens, then you lower the activity of phosphofructokinase 1, and you actually increase the activity of the gluconeogenesis enzyme, of fructose 1,6-bisphosphatase, and then you get lowering of glycolysis and increased gluconeogenesis. This whole page, this page 260, remember, occurs in the liver. This occurs in the liver so that the liver can now secrete glucose. It's a beautiful system. Just depending on where you phosphorylate phosphofructokinase 2, that determines whether it's going to be active or inactive, just that phosphorylation site, okay? A serine in one place gets phosphorylated in the phosphofructokinase 2, and a serine in another location gets phosphorylated. Okay, we could talk about insulin and epinephrine, but we're going to wait on those. I just want this whole glucagon thing to kind of settle in your minds. Any questions about glucagon? Okay. So we are ready now to talk about the Krebs cycle. So I know that we're a couple days late getting to this, but we did talk about the step that just precedes the Krebs cycle, and that's where pyruvate gets decarboxylated to form what? What does it get decarboxylated to form? Inside the matrix of the mitochondria. I don't remember. I know we talked about a couple of lectures ago. Huh? Acetyl-CoA. Wonderful. That's exactly right. So acetyl-CoA is going to be our beginning point for the Krebs cycle. By the way, I may use the term tricarboxylic acid cycle or TCA cycle, and I may also use the term citric acid cycle. All of those are the same. Krebs for the guy who discovered it, tricarboxylic acid because there are 3 carboxyl groups on some of the intermediates in the Krebs cycle. That's why it's called the tricarboxylic acid cycle. And then it's also called the citric acid cycle because citrate is the first product that's made in the Krebs cycle. So let's go ahead and write out what is the overall reaction for the Krebs cycle. So here's the Krebs cycle. Okay. Where is the Krebs cycle occurring in the cell? Where? Not the cytoplasm. In the mitochondria. Okay, now let's get really specific. What part of the mitochondria? The matrix. Very good. All right, the matrix. Okay, so here we got acetyl-CoA plus 2 H2O plus 3 NAD plus FAD plus ADP plus inorganic phosphate. Those are all the reactants. And they're going to produce 2 carbon dioxides, 3 NADH plus 3 protons plus FADH2 plus coenzyme A plus ATP. Okay. So that is the Krebs cycle. How many carbons are in an acetyl group? How many carbons are in an acetyl group? Not 4, not 3, acetyl group. Acetate? Not 1. 2. Okay, so there are 2 carbons in an acetyl group. Guys, memorize the structure of acetate. Okay. Okay, so there's 2 carbons in an acetyl group, okay? And those 2 carbons are eventually going to be oxidized to 2 CO2s. Where are the electrons going to? You know that electrons have to be getting out of the acetyl group, okay, in order to be oxidized to carbon dioxides. Those electrons go into NAD plus and FAD, okay? So these are the oxidized cofactors, okay? And these are the reduced cofactors that I'm underlining right here. So they become reduced. That's going to be key, because if you remember my overview many moons ago, these are going to be used to feed into something called the electron transport chain, okay? This NADH and FADH2 in a process known as oxidative phosphorylation. So keep your mind on these cofactors. They are important, okay? The other thing that's going to happen here is that we are going to have ADP plus inorganic phosphate be converted directly, darn it, be converted directly into ATP from the substrate. Does anyone remember what the name of that phosphorylation is called? Substrate something. Not substrate level. Yes, I'm sorry. Substrate level phosphorylation. You're exactly right. I just didn't complete the words. Sorry about that. You're exactly right. Substrate level phosphorylation. So we do have, at one point in the Krebs cycle, substrate level phosphorylation. Okay, let's get started on this. So this is a circle that some people call the circle of death, but no, it's not. This is the Krebs cycle, okay? And we're starting off here with acetyl-CoA, and in the first step, we're going to make citrate. We're getting 2 carbons from this acetyl group right here, and they're combining with the 4 carbons of oxaloacetate, and together, they're going to make citrate. And I want to point out clearly here that you can see citrate. How many carboxylic acids does it have? Has 3. That's why it's called also the tricarboxylic acid cycle. You guys see that? Okay. We're going to have a dehydration, which is very simple. We're going to just get rid of an H2O, and we're going to create a double bond right here. And then we get isocitrate to be formed. Okay, so we get basically a rearrangement here. This OH up here in citrate is going to end up over here in isocitrate. And then we go from isocitrate to alpha ketoglutarate. And here I want to emphasize that we are oxidizing. So here we get CO2 out. We also get NADH out here. Okay, so the electrons are going to get into NADH. Alpha ketoglutarate is going to go to succinyl-CoA. Here's another position where NAD plus gets converted into NADH, and we're going to create CO2. That's step number 4. And then step number 5, this is where we have our substrate level phosphorylation. Okay, so we have, to be honest, the actual substrate is GDP, but you can think of GDP and ADP as being equivalent, okay? Because essentially the GTP can be converted to ATP. There are transferases that can do that. But the actual substrate is GDP, and it gets converted to GTP, which counts as an ATP. And then finally, I shouldn't say finally, then we have another step here, step 6, where we have succinate going to fumarate. And in this case, we have FADH2 being produced, okay, which is another reduced cofactor. Fumarate going to malate. That's dehydrogenation, no oxidations going on here. And then the final oxidation step is where we go from malate to oxaloacetate, and here we produce NADH. Many of the oxidation steps are carried out by what are known as dehydrogenases. Okay, so that is the Krebs cycle. Let's go ahead and go through the first reaction here. The first reaction is where we have acetyl-CoA. And you can see they're only showing the business end of the cofactor, coenzyme A, bound to the carbonyl carbon of the acetyl group. And that's going to attach to the oxaloacetate. And also, you need water to do hydrolysis to remove this coenzyme A. Okay, so we're going to make citrate here. How does it work? Acid-base catalysis. So we are getting used to this acid-base catalysis, hopefully. Okay, this is on page 605 of your book. Here we have an aspartate, which acts as a base here to remove this proton. And we're going to make a double bonded carbon here. And in order for that to happen, you have to have a concerted reaction where the ketone gets converted into an alcohol, and that's going to be donated by, the proton is going to be donated by the histidine. Okay. And then we've got this reaction where the histidine comes back and now removes that proton that it just puts on, and you break this double bond to the double bonded carbon, and now one of those bonds are created between the alpha carbon of the oxaloacetate, okay? And now you're going to create an alcohol. And then finally, you're going to have H2O come in and remove the coenzyme A, and that's going to recreate the coenzyme A, and now you've got citrate. So citrate is going to have these 6 carbons, and actually this is quite nice because they actually have in red for you where the acetate came from. So these 2 carbons right here came from the acetyl-CoA. Okay. All right, so that is citrate synthase. Citrate synthase. Okay. Next, we've got an isomerization reaction occurring here. So in the next step, we're going to go from citrate to D-isocitrate. Okay. And we're going to remove a water from this citrate. Does anyone know what this symbol means? This little 3 thing here? Does anyone know what that means? Tertiary. Good, 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 good. Well, what's so tertiary about? What does it mean by tertiary alcohol? Anybody know? Very good. Which alcohol is bound? Oh, I guess that's the only alcohol there. The single alcohol there is bound to a carbon, which itself is bound to 3 carbons, right? Right. So we got this alcohol, then you got this carbon here, and it has 3 bonds to carbon. 1, 2, 3. If this had been a 2, that would have been called a secondary alcohol. And if it had been a 1, it would have been a primary alcohol. Anybody know a name of a famous primary alcohol? Methanol's good. Ethanol, probably the most famous, right? So ethanol is probably the most famous primary alcohol. Good, 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 good. Okay, so you guys got your organic chemistry down. Oh, I guess they made here a secondary alcohol here from this. Okay. Yeah, don't worry about this mechanism here. That's a little bit too complicated. You got things flipping over. I can't. That's hard to follow. Okay. By the way, for this for citrate synthase, by the way, they've got this inhibitor called. Oh, I'm sorry. Let me just widen this up for you so you can see what I'm talking about. Called fluoroacetate. So fluoroacetate is very good at inhibiting. Actually, it gets converted into 2-fluorocitrate. So this acts just like acetyl-CoA, except that once it gets to the 2-fluorocitrate stage, it inhibits iconitase. And this is actually a very strong inhibitor of the Krebs cycle. Okay? So this fluoroacetate, it's pretty poisonous. The next step is going to isocitrate dehydrogenase. And when you see this word, dehydrogenase, what class of enzymes does it belong to? It belongs to oxidoreductases. Dehydrogenases are oxidoreductases. Okay? So here we go from isocitrate to alpha-ketoglutarate. Your book messed up here, so I want you guys to make a change in your book. That should be a CO2, a carboxylic acid here on this position. I think they just wrote 3 O's for some reason. Okay, how does this work? You have a base that comes from the enzyme. It's going to remove that proton from the alcohol. You're going to create a double bond between that oxygen and that carbon. And then you've got this hydrogen with 2 electrons leaving. What's the name of a hydrogen with 2 electrons? Hydride. And so we call this process a hydride transfer, right? So we got hydride transferring onto the NAD+. You guys can imagine the ring in that NAD+, right? And you can imagine the hydride attacking the top of the ring, and then there's a concerted reaction where now the nitrogen, which was previously positively charged, is now going to be neutral, right? You can see it in your mind, right? Because I went through that. Okay, and so that's called exactly. It's a hydride transfer. Now we have reduced NADH. So NADH is now is the reduced form. Okay, and now we have oxaloacetate being made, and then you have decarboxylation occurring. So you get this carboxyl group that comes off. And now you've created alpha ketoglutarate. Okay. All right. And when this gets decarboxylated, you get protonation at this carbon right here, at this alpha carbon. Okay. Next step. Here is a case where you've got alpha-ketoglutarate going to succinyl-CoA. And I want to mention a couple of things to you. It mentions here that this requires NAD+, coenzyme A. I want to mention a couple of other cofactors that are involved. There's another cofactor called TPP. there's another cofactor called lipoamide, okay? And there's another cofactor called FAD as well. I have listed here 5 cofactors that are involved in this reaction. Do you recall another reaction where I use 5 cofactors like this? Someone said it? Pyruvate dehydrogenase complex, right? That monster enzyme that has E1, E2, and E3 in it, right? And so this enzyme acts exactly the same as pyruvate dehydrogenase complex. Does anyone remember what the substrates are for pyruvate dehydrogenase complex? What are the substrates for pyruvate dehydrogenase complex? Pyruvate, okay, so pyruvate's one of them, okay. And then there's a cofactor called coenzyme A, right? And if you recall, you get actually a removal of one of the carboxyl groups from the pyruvate. Well, it's exactly the same thing happening here. Alpha ketoglutarate is going to give off CO2, okay? You're going to get NADH created at the end, and you're going to create succinyl-CoA, okay? This is a very, very similar to the pyruvate dehydrogenase complex. Same cofactors are involved, okay? Okay, we're now ready to do our substrate level phosphorylation.
Can I just ask you what you wrote there?
Right here?
Yeah.
I'm sorry, it didn't come out very clear. Let's see if I can see. So I wrote TPP, lipoamide, and FAD.
So they exactly the same cofactors?
Same cofactors, slightly different enzymes.
Okay.
Slightly different enzymes. Those are the that's the difference. But it's going to be the same concept where enzyme 1 starts off the reaction, then takes its product and hands it off to enzyme 2, then enzyme 2 hands it off to enzyme 3, and then you get the product made. Okay? All right. Next step is this substrate level phosphorylation. Here you have succinyl-CoA plus an inorganic phosphate. And again, they're kind of exchanging this. It's going to be GDP. And then that's going to create succinate, ATP, and coenzyme A. How does it work? Let's see if this is a good, easy to understand mechanism. So here's succinyl-CoA. You get phosphorylation. Okay, let's see if we can do this. So phosphorylation. So here I got, I'm sorry that my pencil is not coming out. Oh, that came out good. Okay, so I guess it depends on the reflection. So that's inorganic phosphate. That's going to make an attack right here. That sounds reasonable. Okay. So then we create that. Okay. And so now this coenzyme A is going to leave. We got the phosphate on here. And in the next step, we have this enzyme that has, again, a histidine as a part of the active site. That nitrogen on the histidine is going to attack this phosphoryl group. And I want you guys to really pay attention to my arrow pushing, okay? I'm trying to be as specific as possible in terms of where it's going. I'm not saying it goes in the general direction. I'm actually saying it's going exactly to that atom, phosphorus. So when you guys do arrow pushing, make sure, obviously, you have to be attacking an electrophile, okay? Secondly, be very specific about where your arrowheads are, okay? I'm using a double arrowhead here because 2 electrons are moving. On some occasions, we'll use a single arrowhead. In other words, actually a half an arrowhead if we're just pushing around 1 electron, okay? So I'm trying to be very careful here, okay? And then this bond gets broken, and these electrons are going to end up on this oxygen. Okay. All right. So then we've got this N-phosphohistidine, and then finally we get GDP, actually, that's going to attack this phosphoryl group, and you're going to make GTP from this. You guys should know this mechanism, right? Basically, I'm going to take, this is going to be the beta phosphorus from GDP is going to attack right there on that phosphorus atom, and then this is going to attack a proton. Okay, it's going to attack a proton. Okay, and then we're going to get GTP made from this. Okay. Next step. We're going to go from succinate plus coenzyme Q, and we're going to make fumarate and reduced coenzyme Q. This is the same coenzyme Q that you guys can walk into one of those nutrition stores and purchase as CoQ. So CoQ, you probably have heard that term CoQ, right? So it's the exact same stuff. And it actually occurs not directly, but through this cofactor that we've discussed before called FAD. And FAD is composed of riboflavin, important vitamin, okay? But this, I've circled here the business end of FAD. So I've done this before. This is the business end of FAD. And so this FAD is going to help carry out this reaction. Let's go ahead and do this. Succinate, fumarate. Whoa. So succinate that is the reaction we want to carry out. Succinate looks like this. And we have this business end of the molecule. Whoa. That's going to look like this. That's the business end of the molecule of FAD, okay? And so what's going to happen here is do this like this. So here I'm drawing an arrow with just a half an arrowhead. Is it time? Yeah. Okay, we're going to come back to this next time, but I just want to let you guys know that we're going to get one electron movement plus the proton. And so that's going to be the mechanism here. And so what's going to happen is we're going to create a double bond here, and we're going to get, actually I better finish this up before I forget. And so we will get 2 more hydrogens, 2 hydrogens added on to the FAD, okay? And also, we're going to get fumarate. Okay, I'll repeat this next time. I hit the eraser button. I don't know how to undo that. I should be able to undo that, but okay. We'll do that next time. I'll hand you guys back your quizzes. The high was 20, and the average is about 13. I also want to mention, now that I'm thinking about it, that I understand that sometimes my style of lecturing and testing might be different than others, and so I look for improvement in your scores as we proceed through the quarter. And if that happens, I'm willing to de-emphasize the first midterm and the first quiz or two, okay, and emphasize what you guys got at the end, okay? If you guys go the other way, go down, then I won't do that. I'll just take straight points, okay? So there is a chance, there's hope, okay? Hope with a capital H. There's hope for those of you that didn't do well on the first midterm, but are doing better now as we proceed, okay? As we proceed to the second midterm and the final, I will bump up your grade if you're on the border, if you continue to progress all the way to the final. Han. Francisco. Yvonne. Mindy. So when you get done looking at your quiz, you can just bring it up. Make sure I tally everything right. Alejandra.
Which one?
Tejeda. Derek. Andy. Prenny. Vanessa. Paul. Paul. Carlos. Carlos.
I'll give it back, right?
He's right here. Thanks. Gretchen. Claudia. Gwen. Gwen Law. Audrey. Omar. very straightforward.
easy.
Yeah, yeah.
By the way, for the midterm, what should we be concentrating on? Like, just everything?
Everything.
Steps and everything?
Steps, anything that I went over.
Okay.
Yeah,
Okay, good.
yeah. It's gonna be
So it's more difficult.
it's gonna be more difficult.
Yeah.
It's more material.
Yeah, right. Okay.
I
thank you.
agree. Moving on. Maria.
This sometimes it does take longer.
You know, there's
Because I was.
it's got to at some point. So it either does it after a tax year, and then it could do it, or it could do it right now. I think it's probably this. I think it's probably correct.
Okay.
This makes the most sense to me.
Okay. Yeah, because I wasn't sure about that. Whether it came before or.
And there's probably some debate on the issue,
Okay.
but this is reasonable. If it's reasonable, I accept it.
Okay. Thank you.
Mm-hmm.
So my uncle just passed away and I have to go to a funeral next Friday
So
to
sorry.
Fresno.
Oh my gosh.
So I
Is
don't,
Friday special? What's happening on Friday?
I'm not sure. That's why I haven't really checked, so
Let me check. What's happening on Friday? I think I have my syllabus here somewhere. Be patient with me. No, our midterm's not until May
Monday.
9th.
Oh, okay.
So you said you're gonna be gone on the 6th, right?
Yes.
Okay, you'll be gone on the 6th, but we will have the midterm on Monday.
Okay, that's fine.
Yeah. All right. Good luck. I'm sorry about your uncle. Was it sudden?
Like 3 weeks. They told me I had terminal cancer and he just passed away.
So fast.
Yeah.
So fast. I hope he wasn't suffering. Was he older?
He was 60, so he wasn't that much older.
God, that's scary.
Yeah, he went into diabetic coma on Sunday and then like 3 days later on a Saturday he just passed away.
Ah, painful, painful. I'm sorry. Well, my condolences to you and your family.
Okay.